Well, good afternoon, everybody. Welcome to the second virtual edition of the DOE Isotope Program Lead-212 user meeting. Thank you for joining us today from wherever you may be and whatever time zone that you may be on. For those of you that don't know me, my name is Roy Copping. I'm a group leader at Oak Ridge National Laboratory. My group is responsible for the production of a variety of isotopes with applications in nuclear medicine, including radium-224, Lead-212 generators. At this point, I really want to thank the DOE Isotope Program again for inviting me to moderate this session again. I think we have a fantastic program for everyone's viewing pleasure again today. Following a brief introduction from me, providing a very brief historical perspective from isotope production at Oak Ridge and our current radium-224 Lead-212 generator. We'll then move into our selection of fantastic speakers who are currently working in the field. We have Mengshi Li from ViewPoint, Rebecca Abergel from the University of California, Berkeley, Ebrahim Delpassand from RadioMedix, and Matt O'Hara from Pacific Northwest National Lab. Following the talks, we'll have a moderated Q&A segment, and at this point, I'll just remind everybody to please submit your questions to the Q&A tab that you'll find at the bottom of the meeting, and we'll do our best to answer those questions at the end of the talks. So to save all the time for our fantastic speakers, we'll move quickly into my talk. And I'm going to provide, like I said, a little bit of an introduction on the radium-224 Lead-212 generator that we're currently producing from my group at Oak Ridge. But I just wanted to give a very brief historical perspective of isotope production at Oak Ridge. And to provide that historical perspective, we need to go all the way back to the 1940s. For those of you that might not be aware, Oak Ridge has been engaged in isotope production since the 1940s, where following the Second World War, the calutrons that were previously used to enrich the actinides were turned to enrich stable isotopes as well, and the stable isotopes that were enriched then be in turn radiated at the graphite reactor that was also at Oak Ridge, and then shipped to customers and researchers for their use in applications in nuclear medicine. And the first isotope that was shipped from Oak Ridge in 1946 was carbon-14, and then since then, Oak Ridge has a very rich history in the field of isotope production, with a few key points in history that I'll just allude to. As I just mentioned, in 1946, Oak Ridge made the first carbon-14 shipment to a hospital in St. Louis. And then later in the 1940s, Oak Ridge started the large-scale mouse genetics project to study the effects of radiation on mammals. It lasted all the way to 2009. Another key point in history from an isotope production perspective of Oak Ridge was in 1993. Here, where you'll see a photograph of researcher Russ Snap working on a tungsten-188, rhenium-188 generators, which of you are probably very familiar with. Also, in the 1990s, Oak Ridge established the production of actinium-225, that we all know has important applications in targeted alpha therapy treatment. Later in the 1990s, the Office of Biological and Environmental Research started the low-dose radiation research program at Oak Ridge that lasted all the way to 2011. And then more recently in 2017, a key point to mention is Oak Ridge had a production contract with biopharmaceutical to provide actinium-227, and that actinium-227 is used to provide radium-223 that's currently used in the FDA-approved drug Xofigo for the treatment of metastasized prostate cancer. But getting back onto the topic of this user meeting today, that's Lead-212, and I just wanted to highlight some brief points about radium-224 Lead-212 generator production at Oak Ridge. This was a production effort that was established back in 2014 at the request of many customers. For those of you that may not be aware, radium-224 and Lead-212 are part of the uranium-232 decay chain, and radium-224, with a half-life of 3.6 days, can be milked periodically from its long-lived parent, thorium-228, that has a half-life of 2 years. And this radium-224 can then be loaded on a generator, and the generator can be milked periodically for its daughter Lead-212, has a half-life of 10 hours, and it can also be milked for its other daughter, bismuth-212, for use in nuclear medicine applications. My group is currently providing generators up to 20 millicuries. Typically provide generators every 3 weeks, and we have a large stock of thorium-228 available to us that we can prepare the generators from. This is a photograph of our generator that we prepare. As you can see here, the generator is composed of a very small amount of cation exchange resin in this small plastic tube. Radium-224, after it's been milked from thorium-228, is loaded on the generator. This generator is packaged in a lead pig, and the Lead-212 can be milked periodically from this generator using hydrochloric acid. This is the generator set up in a stand, just to show you how it's used. And this is the elution profile of Lead-212 from that generator. And the Lead-212 can be milked from the generator with a few bed volumes of 2 molar hydrochloric acid for use in a variety of research applications. So that's the brief introduction that I wanted to give on radium-224 Lead-212 generators. From here, we'll move into our speakers for the day. First of all, we have Mengshi Li from ViewPoint to kick us off. So if you can start sharing your screen, Mengshi, and then take it away. Thank you.
Well, thank you, Roy. Hi, this is Mengshi Li. I'm the principal scientist at ViewPoint Molecular Targeting here. I would like to thank the organizers for putting together this workshop. Lead-212 has been a very important part of our technology, and we're especially interested in the Lead-203 and Lead-212 based theranostics. Today, I'm going to talk about some of the work we've been doing to better understand the dosimetry and the renal toxicities using these pair-matched isotopes. Here are my disclosures. So I'm going to start with the radio labeling of both Lead-212 and bismuth-212, and then talk a little bit about the dosimetry work we've done for the Lead-212 labeled peptide, and then I'm going to introduce some of the data we found, and for the very interesting biomarkers for acute kidney injuries, including Abergel, TIMP2, and IGF-BP7. Specifically, the TIMP2 and IGF-BP7 are FDA-approved assays for AKI. They're called nephrochecks. And then I'm going to talk about the histopathology analysis for the kidneys, in the kidneys. So I'm sure you're all pretty familiar with the decay scheme within the radium-224 generators. Along with the decay, polonium and thallium are not expected to stay in the chelator, whereas the stable chelation of Lead-212 and bismuth-212 are fairly important. 238 keV gamma from gamma emission can be used to monitor the Lead-212, whereas the 583 keV gamma emission from thallium can be used to indirectly monitor the bismuth-212. So the radium-224 generator is eluted by hydrochloric acid, and that contains all the decay daughters. And using 2 different resin, we can selectively purify Lead-212 and bismuth-212, including the lead resin and the true resin for bismuth. And putting those two resins together, we can get purified Lead-212 and bismuth-212 at the same time. So to test the radio labeling, we use the chelator conjugated cyclic peptide BMT01. And specifically in this case, we use the less specific chelator with the amide at the diagonal position, which can provide a neutralized charge for bivalent metal like lead. So we started with a purified lead. The radio labeling reaction was conducted at 85 degrees for 15 minutes, and the radiochemical purity was determined by thin layer chromatography using sodium acetate and DTPS mobile phase. As you can see here, all the lead was incorporated into the reaction. We cut different portions of this TLC strips to monitor both lead and bismuth. And after 3-hour incubations in human serum, we start seeing a little bit bump in the upfront for lead-212, indicating there might be some decoupling of lead-212, but generally that's less than 3%. And we also monitored the bismuth-212 that was being generated from the lead-212 decay. In this case, we didn't really actually find any ejected bismuth upfront here, but after incubated with the in the serum for 3 hours, we start seeing a little bit ejected bismuth upfront. That's still less than 3%. So moving on, we use the true resin actually to purify bismuth, because we want to test how bismuth itself labels the peptide. And we introduced the bismuth into the reaction, and under the same reaction condition, we found most of the bismuth it was incorporated into the peptide. And then we moved on to have both lead and bismuth introduced to the reaction at the same time. So with the coexistence of lead and bismuth, lead was still successfully incorporated into the chelator. However, with the interference of lead, you start seeing more unlabeled bismuth, which is about 7%. So generally, we found we found both stable chelations for lead and bismuth in this peptide. So moving on, we did the biodistribution of lead VMT01 in the CD1 mice using the Lead-203 surrogate. And not surprisingly, this and monitor the bio-D for up to 55 hours. Not surprisingly, this peptide was cleared majority through kidney, bladder, and urine. So kidney in this case was the dose-limiting organ for both male and female mice. And our colleague Dong-Yo Li helped us to do a dosimetry analysis for the lead-212 therapeutic counterpart using the particle and heavy ion transport code system in the DigiMouse software. And we found that alpha particle contributed to 93% of the total dose exposure in kidney, whereas the electrons contributed to slightly above 7%. So in these CD1 mice, we injected 0.9, 3, and 6.7 megabecquerels of lead-212 labeled VMT01. The dose in kidney ranges from 1.9 to slightly under 14 gray, based on the dosimetry analysis. And we monitored those the weight all the way up to 7 months, and we didn't really observe acute toxicity. But later on, there was a differentiation in the weight, especially for the higher dose groups. And throughout the process, we collect the urine and blood to analyze the kidney toxicity and blood hemotoxicity. Specifically for the earlier stage, when we analyze urine biomarkers for like Abergel, TIMP2, and IGF-PP7. So interestingly, we found that as early as the first day post-injection, we saw a huge increase in the urine Abergel. And later on, during the transition phase, it was the parameter, the biomarker went back down to background. There's no difference between the control and the dose groups. However, at the late stage, we saw the Abergel concentration increased back again. And in this case, the increased concentration of Abergel was only seen in the medium and higher dose, and it was not seen in the lower dose group. And we also found that at the earlier phase, the TIMP2 and IGF-BP7 was also increased in the lead-212 VMT01 dose groups. So those results demonstrate that these biomarkers are fairly sensitive at the earlier phase, especially for the acute kidney injuries. And the blood chemistry analysis indicates that there was no liver toxicity. However, at the later time points, the blood urine nitrogen and creatinine concentration increased, indicating that there was a kidney functional loss. However, these biomarkers are not sensitive at the earlier phase. So we also did the CBC analysis, and we found a decreased red blood cell concentration and hemoglobin concentration that's probably associated with the kidney damage, which caused the anemia. So our colleagues, Perna Restology, help us to do a histological analysis of all these kidney slides and score the injuries for different parameters, including tubular damage, glomerular damage, histological inflammation, and fibrosis. And we found a dose-dependent increase in the scoring of tubular injury and glomerular changes. However, the damage appears to be more concentrated in the tubular area rather than the glomerular area. This could be explained by the short path length of the alpha particle. Again, the dosimetry analysis indicates that the alpha particle contributed to 93% of the total dose. So more dose was more localized in the tubular area rather than the glomerular area. These histological changes were came along with this inflammation and histological fibrosis. So putting together, we found a stable chelation of both lead and bismuth in the chelator, and kidney dosimetry for Lead-212 therapeutics was established using the Lead-203 surrogate, and we found a fairly interesting result in the earlier phase, where the urine biomarkers like NGAL, TIMP2, and IGF-BP7 were very sensitive. However, the blood chemistry biomarker like BUN and creatinine were only sensitive for the later stage. And histological staining indicates that there were tubular damage and glomerular damage with a more concentrated damage in the tubular area. So generally, more detailed understanding are needed to understand the toxicity that's associated with the radiopharmaceutical, especially alpha particle therapies. So with that, I would like to thank all my colleagues here at ViewPoint and also at the University of Iowa, and also the Diana Zapata from Nationwide Children's Hospital, who helped us to do all these kidney assays. And of course, Oak Ridge and DOE for providing the generators and isotopes for us. Thank you.
Thank you very much, Mengshi. So any questions for Mengshi, please submit them to the Q&A thread, and we'll try and answer them during the moderated Q&A session at the end of the presentations. So moving on, we now have next have Rebecca Abergel from the University of California, Berkeley. I don't know if you want to go ahead and share your screen, Rebecca.
All right, great. Thanks. Thank you, Roy. Hi, everyone. So I guess I'm a little newer to the use of Lead-212 for targeted alpha therapy, but what I was going to show you today is a small case study we've done for an industrial sponsor recently, and we're looking at melanoma growth suppression in a mouse model. And so first, I just wanted to give a little bit more background. Maybe everybody's familiar with this already, but this is always good for myself as a reminder. Those are the elements that we're interested in for isotopes that have potential in the field of targeted alpha therapy. And what's we're focusing here on is Lead-212 and the in situ generation of bismuth-212 as an alpha emitter. But I guess what I wanted to highlight is that lead and bismuth are metallic isotopes. And lead sits kind of in the sweet spot in the list here. I've listed them in terms of the half-life of the, I guess, the generator, the isotopes that would be injected in a treatment. And so we're going from above a week in terms of half-life all the way to radium-223, and astatine and bismuth-213 are from less than an hour to a few hours. And then so lead is a bit longer than this, and it's actually very helpful in terms of treatment formulation and handling the isotope itself. So that's really an advantage that I think we've, at least we've seen during this study that allowed us to look at a study design that was pretty extensive. The other property that is interesting again is that it's a metallic isotope, so we have access to a bunch of different chelators for linking or radiolabeling biological vectors. And in this case, we're looking at antibodies, and so we have to be careful in terms of the metallation conditions. We cannot heat things too high in terms of the temperature or for too long, otherwise it will destroy everything. And so this is one thing again that has been fairly easy in the case of lead and bismuth in comparison to everything we've done in the past with other isotopes such as actinium-225. So here is the study design, or at least part of it, that we were working with. So again, we're looking at a mouse model with melanoma cancer. So the cell line that we're using grows really fast. After inoculation, we had tumors appearing at about 3 days on the order of 100 cubic millimeters, so ready for treatment. And then we're looking at different treatment regimens from a single dose at a fairly low dose of 10 microcurie per mouse, all the way to a single dose at a really high level, 40 microcurie per mouse. And then also 2 consecutive treatments, 1 3 days following the tumor inoculation, and then the second one 10 days following the inoculation. So to see if there's any effect from not a fractionation regimen per se, but at least a doubling the treatment. We have controls. I'll show you controls, which is the antibody themselves at a level that is equivalent to the highest dose that we've used. But what was interesting in this study is that we wanted to compare it to bismuth-213, which is kind of the more accessible, most comparable isotope that we had in this case. And so again, same idea. We have either a single dose or a 2 dose at much higher activity levels, just because bismuth-213 has a shorter half-life, and we just need to go a little higher in dose. We had non-targeted treatment. We had a cold antibody control. So pretty standard design. So in terms of the comparison, we're looking at Lead-212 versus bismuth-213. And so we did get different generators, the radium-224 generator and the actinium-225 generator, both from Oak Ridge. Again, I would highlight that in this case, working with the Lead-212 was much easier in terms of just because of its half-life is much longer. And so injecting a lot of mice with a treatment that has Lead-212 is much easier than with bismuth-213 and a half-life of 46 minutes. So we had different generators to work with in parallel. And then here is the elution profile from the Lead-212 elution, which if you've paid attention to what Roy shows is a little bit different from what we were expecting. And so I think this also kind of, it was the first time we were handling a Lead-212 generator. And so, I would advise anyone who's starting with this to run an elution profile on the day of receipt before the experiment, just because there are some surprises. But in the end, we did get all of the activity we wanted out in a fairly dilute HCl water mixture. We also followed the elution of thallium-208 and bismuth-212 at the same time. All of this is counted on the high purity germanium detector. And what we took from these fractions here, so all of the lead we eluted from the generator was then concentrated or exchanged it to concentrated nitric acid and then back into dilute HCl, just to make sure we didn't have any other contamination from the resin itself, and to make sur we had the adequate concentration to move forward. This takes about 3 hours in terms of the matrix exchange itself. And then we did use different chelators. Those are pretty standard again, but for lead, we used a TCMC that can be conjugated to the antibody. And then for the bismuth, the Chexe DTPA that can be conjugated to the antibody. So we have, in terms of the characterization of our antibodies and radio-labeled antibodies, we have either 2 or 3 chelator antibody ratio. So it's pretty well determined using MALDI mass spectrometry. And for the radio-labeling procedure, again, those are pretty standard. We are in ammonium acetate mostly for both bismuth and lead. And we do things at 37 degrees for an hour maximum. And we've seen pretty high purity and pretty high radio-labeling yields for the lead. And so if I'm showing you here what we saw for different elutions that came over time after receipt of the generator. So we do see the radio purity going down a little bit. The radio purity is determined by ITLC. We went from almost 93% to a pretty low purity of 28% after about 2 weeks, with several elutions. And the labeling yield went for about 85% to 50% as we went over time. So the generator is very reliable for the first week, but then it becomes a little bit more difficult to extract a lot of lead for pure radio labeling. And so again, back to our study design, I'm just going to show you the results for the initial doses here. So we're going to compare a single dose of Lead-212 versus 2 doses with a single dose of bismuth versus 2 doses. So please note the difference in activity levels. We injected 10 microcurie per mouse for lead, 200 microcurie per mouse for bismuth. And those are the corresponding survival curves. So our controls here, again, we're injecting treatment at about 100 cubic millimeter for our tumors. We do have different euthanasia criteria, but nothing went above 1,500 cubic millimeters. We did see we didn't see tremendous toxicity here in any of those groups. So mostly it was tumors growing that led to sacrificing the animals. So a control group here is the survival curve goes down very quickly. The bismuth groups are down here in yellow and purple, and you see that the lead groups both really extended the lifespan of the mice quite dramatically. And so again, this is after either a single or 2 doses spaced 3 days and 10 days apart from the treatment in the tumor inoculation. And so this really demonstrated how Lead-212 could be very efficacious compared to bismuth-213 at this point. So there are a few takeaways, at least from people running the experiments in my group, is that it was much easier to work with Lead-212 than bismuth-213 just because of the longer half-life. We could use much lower doses. The only thing to watch out for is the thallium-208. We could use smaller volume of the solutions of the isotopes. The labeling reactions were easier with higher radio purity and better yields. So in general, it was just all more advantageous to use the lead. And so with that, I'll conclude. The people who did the work for this experiment are mostly Matt Flick and Joshua Chen and grad student Tyler Bailey. The postdocs, Jen Walker, Alyssa Geiser, and grad student Kathy Shields helped with the elutions just because we had a lot going on at once, and Dalia supervised the whole study. And so with that, I'll thank our radiation protection program that helps us dispose of the generators, as well as the molecular foundry at Albia, where we do our MALDI characterization, the NIDC for providing the isotopes, and as well as the Department of Energy. All right.
Thank you very much, Rebecca. Fascinating talk. I'll have to follow up with you at some point about the elution profile from the generator, but we can do that later. So moving on, we'll address any questions that you have to Rebecca at the end of the talk. Sorry, but now let's move on to Ebrahim Delpassand from RadioMedix. So if you want to go ahead and share your screen, Ebrahim.
Well, hi everybody. I'm Dr. Delpassand, Chairman and Medical Director of Excel Diagnostics and CEO of RadioMedix. I would like first to thank the organizer of this meeting. This is our, I think, third year participating in this program. I would like to thank the organizer, NIDC and DOE, for putting together this exciting program. This is a pleasure to provide to you the results of our targeted alpha therapy program that we have for treatment of neuroendocrine tumors. Previously, last year, I provided some information and report on this clinical trial, which was ongoing, and now I'm pleased to provide you more information on the completion of the dose escalation trial and where we are right now, and showing some exciting cases that we have. So this clinical trial was a phase 1, first in human, a non-randomized open-label dose escalation trial. The sponsor of this trial is RadioMedix. We have our business partner and manufacturer, Oranomed. And the clinical trial site is Excel Diagnostics and Nuclear Oncology Center located in Houston. The primary objective, like any phase 1 trial, was to assess the safety and dose-limiting toxicity of ascending doses of alpha-medix or Lead-212 labeled dotamtate. The secondary objective was to look at the pharmacokinetics, dosimetry, and preliminary effectiveness using this drug in patients. Altogether, we have treated so far 33 patients with biopsy-proven unresectable or metastatic somatostatin receptor neuroendocrine tumors. And they are from a different primary site, as I'm going to show you later. So we did the imaging for evaluation by RECIST criteria using CT or MR. We also did F18 FDG PET CT at the baseline, and if any of the patients who had positive FDG, we also repeated the FDG PET CT for the follow-up and evaluation of response to therapy. All patients, they received somatostatin receptor PET CT, whether gallium-labeled dotatate or copper-labeled agents or other FDA-approved imaging agents for the process. We did it at baseline and periodically before each cycle and after completion of the treatment protocol. We also used some tumor markers such as chromogranin A, serotonin, gastrin, insulin, pancreatic polypeptide, and neurospecific enolase. Also, we used liquid biopsy test, NET test, supplied by company REN for evaluation of at baseline and response to therapy. All patients, they also had ECOG performance score and quality of life questionnaire. So I want to just show you some of our examples in here for some of you that you watched my presentation last year. Some of these slides are actually repetition, but I thought it's a good way to set the stage for sharing with you our new results. So this was our first patient treated with the effective dose that we received after completion of single accelerated dose going to multi-accelerated dose, and started seeing efficacy and therapeutic effect. This patient was a 62-year-old man with metastatic small bowel grade 2 neuroendocrine cancer diagnosed in February of 2017. In the subsequent month, he had a small bowel resection, started sandostatin, and then followed by actually left hepatic lobectomy and right lobe wedge resection of the liver, and then enrolled in our trial in April of 2019. As you can see on the image on the left, there are multiple liver lesions as well as a mesenteric lesion pointed in the picture. These are subsequent gallium dotatate images. On the left, we have the first baseline image before the treatment. And as you can see, after 4 injections, which is the current protocol that we are following, and 5-month follow-up after the last injection, you can see the patient is essentially free of disease, maybe very faint uptake in the mesentery, but this was April of last year. As of today, the patient doesn't have any evidence of progression. The next patient is 45-year-old man with history of metastatic lung carcinoid, had metastases at the diagnosis, metastases to the bone and the spine, received a treatment by resection of the lung primary, and later on L3 vertebrae was biopsied, which showed a high-grade neuroendocrine tumor, grade 3. Initially, the pulmonary was G2. He was then started on octreotide therapy and denosumab, and then again showed progression and was enrolled in our clinical trial in early 2019. As you can see, this patient has significant disease in the spine and bulky lesions in the liver. Patient was on 3 types of narcotic pain relievers when we saw him for the first time, and still had a breakthrough pain and had a hard time even laying down on the imaging table. So this patient received 4 cycles of therapy, and as you can see, in March of 2020, this is the gallium dotatate, again 5 months after the last injection, showing no evidence of disease. The patient is basically back to work, and we have a hard time getting him back to Houston for follow-up. So he is doing great with no evidence of progression as of to date. And the next patient, 71-year-old woman diagnosed in 2014, again with bronchial carcinoid. As you can see, almost the entire left lung is involved. A patient was not amenable to surgery because of involvement of great vessels in the mediastinum. Patient was enrolled in our clinical trial. At the time, she was fully dependent on oxygen and was not able to basically have any activity without oxygen, and she was moving with wheelchair. These are subsequent images after 4 cycles, and as you can see, very impressive response to therapy. As of last April of 2020, we can see that there is no evidence of disease, and subsequent even FDG PET-CT at baseline was positive. After 4 injections, you can see there is no residual disease in the left lung. By RECIST criteria, she is considered complete responder. So these were 3 basically all these patients, they were first 3 patients with effective dose. So I'm not showing 3 patients out of 100 patients that, the best pictures to show you. No, these were consecutive patients. And as a result of this impressive response to therapy, we expanded this cohort and enrolled more patients under this trial with the effective and safe dose. And here is the demographic data on these patients. 4 patients with bronchial carcinoid, 2 with PNET, pancreatic neuroendocrine tumor, 2 with rectal neuroendocrine tumor, and 2 with a small bowel. As you can see, the grades are different. One of them was G3, as 2 of them actually G3, and all they had a metastatic stage 4 disease. In terms of response to therapy, I'm pleased to share with you these responses to therapy. 8 out of 10 patients had objective radiological response by RECIST criteria. So we have 80% objective radiological response. By PET, the responses were even more impressive. As you can see, the percentage of effect on the tumor burden on these patients by PET imaging. And you can see the time since first treatment as of June of this year, basically ranging from 9 months to 26 months, and duration of response, again, as of June of 2021, defined as the first time that we saw there is a response by RECIST criteria to last June, are these numbers. As you can see, up to 22 months as of June. So these results are, of course, extremely impressive. In my experience, I mean, I have treated thousands of patients with PRRT, with beta emitters, and obviously these results were extraordinary. And we were very pleased to see these results. This is a Kaplan-Meier curve showing that we have only had one patient progressed, and this is a very interesting case actually. This patient did not progress with somatostatin receptor, expressing tumors. Actually, this patient unfortunately developed non-SSTR positive tumors, and I just quickly present this to you. This was one of our great responders actually. This patient, 50-year-old woman with pancreatic neuroendocrine cancer, as you can see, almost 80% of the liver is involved, and maybe some lymph nodes also in the upper abdomen with the cancer. And gallium dotatate shows highly expressing somatostatin receptor tumor in here. After 4 injections, she achieved complete response to therapy. So this was until May of this year, May of 2021. And here we noticed that patient in MRI that we did on the patient is having few new lesions. This is in the middle, this is the MRI after the last 4 injections, showing complete resolution of all the lesions in the liver. But in May of last year, unfortunately, we saw some new lesions. We immediately repeated the gallium, and the gallium is still negative. We don't see any of these lesions. We did also PET, FDG PET imaging, and for sure we are seeing some lesions in the liver correlating with the abnormalities in the MRI. Subsequently, this patient biopsied on those lesions, and it appears that she has a poorly differentiated or of neuroendocrine tumor again. This is very unfortunate, but at the same time, fascinating case showing that we were able with alpha therapy take care of all the somatostatin receptor expressing tumors, but unfortunately in this patient, a new line of cancer cells are growing, and this time they are not expressing somatostatin receptors, so escaping from our therapy. So this was the only patient this is the only patient so far has progressed, but progressed on a non-SSTR expressing tumor. So we have also started just to test this therapy in patients who are PRRT refractory, meaning that they have received at least 4 courses of Lutathera or lutetium dotatate. We are just trying to learn how our therapy works in these patients, and these pictures are right out of the oven, if you will. Just have a few examples after 2nd injections on these patients. The first one on the top in this image shows almost stable. We start seeing some decrease in the size of the lesions, for instance, but we consider this as stable. This is a 71-year-old man with refractory pulmonary carcinoid, again treated previously in 2017 with lutetium dotatate PRRT 4 cycles, and then progressed, and we tried alpha therapy on this patient. And then the next one, which is actually impressive, after 2 cycles only, you can see these liver lesions in the baseline gallium dotatate or copper dotatate in this case, and no liver lesion in here. By the way, these hot spots in the chest, the patient had a port, and we injected the drug in the port, and so this is the tip of the catheter, and these are some activity in the catheter. So these are not disease in here in the chest. Liver is completely clean in this case. A couple of more examples again. Another one after 2 injections, as you can see, we just start seeing some effect. This one probably again stable case, but this one for sure shows initial effect of the medication and resolution of many of these lesions in the mid upper abdomen and in the chest. So this is just very preliminary, as I said, right out of the oven, and we were excited to show you these images. Another example, we were able to get a compassionate permission from FDA, which later on was approved by IRB at Excel Diagnostics. This was a patient, one of my patients, who had treated with 4 cycles. This was a PD patient. I first saw this patient when he was only 8 years old, and received 4 cycles of Lutathera, and actually responded. The patient was stable until last year. Unfortunately, at that point, last year, we noticed that there are lesions in the liver that now basically suggest progression. You see the baseline gallium before alpha therapy in this patient. And then after 4 cycles, this is such an impressive response in this boy. He is 12 years old. He is going now to summer camp with no evidence of any pain, or which was really debilitating for him, preventing him to go to school or play. And now, as you can see in these blue pictures, probably you can appreciate much better the resolution of all these lesions in the liver. So this was really exciting, and I wanted to share with you the results on this. And this is, as a scientist and as a physician, this is the greatest payday for us. So in summary, this study included dosimetry, included hematological and blood chemistry for safety evaluation, which was done periodically according to the protocol. In summary, 163 adverse events were reported to our CRO, 52% of them from the SAD or single accelerated dose cohort, and 48% in the multi-accelerated cohort. Majority of these were grade 2. I can tell you that none of these were clinically significant, and we didn't have any clinically significant drug-related or investigational drug-related AEs or serious ADs. We also didn't have any dose-limiting toxicity causing de-escalation or decreasing the dose in any of our subjects, and all our subjects tolerated the treatment well. We didn't have to cancel any doses or anything among the cohort that we have tried so far. So this has been our milestones. We jointly with our partner Orano Med filed our patent in 2017. In January of 2017, phase 1 study started in February of 2018. We received orphan drug designation from FDA on October of 2018. We completed the dose escalation of the trial in December of 2019, and we went into expanded cohort and enrolled until August of 2020. Enrollment of the phase 1 PRRT naive patients completed in April. And of course, we are in the follow-up phase, and we will start our phase 2 trial for PRRT naive in September 2021, and also our refractory patients at the same time we will continue. So I think we have done this in a relatively short period of time, considering all the challenges that we have had. One challenge that we haven't had is enrollment of patients. Patients, they are extremely eager to participate in this trial because they have seen the results that we have. And that's one of the major advantages that we have, which, there are many patients who are willing to participate in this trial. So in summary, in terms of efficacy, we have objective radiological response in our PRRT naive group by 80%. This obviously compares to a direct competitor or our, the standard of care, which is available right now for our patients, Lutathera, which has an objective radiological response of only 13%. And I showed you one patient who progressed, and that was non-SSTR expressing tumor. In terms of safety, this drug enjoys a very good safety profile with no clinically significant investigational drug-related renal, hematological, or hepatic toxicity. So in terms of indications, we think that number one, there are some advantages in here. We are not dealing with or selecting any primary site of the tumor. We are basically going by SSTR expressing neuroendocrine tumor regardless of location of primary or even grade Ki-67 index. We think that this is the right way to go. It's based on biology and not lumpology, as is the title of one of our friends' presentations, regarding targeted therapy. The current standard of care, PRRT Lutathera only, is approved for GEP-NET, as we know, and there are certain grade limitation, grade 1 and 2, with Ki-67 of less than 20. There is no limitation for us in the current protocol, and we think it can be included in the indication. And it can be used for first-line therapy for naive patients, as well as hopefully second-line therapy in patients with refractory to lutetium dotatate, which at that point, really, they don't have any other therapeutic options. We are moving now toward phase 2 trial, which will start next month. And as mentioned, enrollment has been extremely quick, and it's going to be even faster when we go to multi-center trial. And our partner, RadioMedix, has expanded its commercial manufacturing bandwidth, and we are looking forward to approval of these drugs so we can make it available to all our patients with somatostatin receptor expressing neuroendocrine tumors, and treat more patients so that they can enjoy the therapeutic effect of this breakthrough medication. Here, I would like to thank all our partners, our team at RadioMedix, at Excel Diagnostics and Nuclear Oncology Center, our business partner and manufacturer, RadioMedix, Rapid Group, that they did the dosimetry for us, InClean, our CRO, and the last but not the least, the help from NIH. I really would like to appreciate all their helps in supporting us for performing this exciting clinical trial. And without their help, it was not possible to accomplish what we have done so far for our patients. And here, all collaborators, all together, we are changing the nuclear medicine landscape from innovation to intervention. And I would like to thank you for your attention to this, and I'd be happy to answer any questions you might have at the QA session. Thank you so much.
Thank you very much, Dr. Delpassand. There are some questions for you, but we'll get to those in the Q&A session. For now, let's go ahead and move on to Matt O'Hara's talk. So if you want to go ahead and share your screen, Matt, and then take it away.
Thank you very much for the opportunity to present today. And Dr. Delpassand, that's extremely exciting news. So thank you for that update. My name's Matt O'Hara. I'm a radiochemist at Pacific Northwest National Laboratory. And I am, I guess, presenting something a little different, which is more or less a progress report on a fluidic remote system that we engineered at the laboratory here, and we're in the very early stages of evaluating its performance. So I'm here to present some updates on that. Okay, so the purpose of why we're doing this is that there is at this point still, I would say, limited generator availability in the United States. NIDC is presently supplying generators up to about 20 millicurie. This is a manual assembly process, which is labor and time intensive. And it also gives a pretty significant dose to the staff members that are preparing these generators. With the increasing demand of this generator, we took a new look at the production pathway. and recognize that this may require improved assembly efficiencies, and importantly, a process that will result in decreased radiological dose to staff. So PNNL has developed a remote automated system to isolate radium-224 from the thorium-228 stock material, and then prepare the radium loaded generator columns. This is all very recent in FY21, and really just within the past couple of months, we've worked up to demonstrating the process up to about 3 millicurie level. And in FY22, we anticipate continued radium loading levels up to clinical levels that would be hopefully reaching 20 millicuries and perhaps up to 30 millicuries in this next fiscal year. Okay, so just a brief overview of the process and system. So it involves 3 fluidically interlinked modules. The first module isolates radium from the thorium-228 stock material. The stock material is injected into the process, and the first step is to remove the thorium onto a column, and then remove the dose, the dose-intensive daughters, and isolate the radium. At the end of that process, the thorium is removed from the primary column and is recycled back for reuse. And at this point, we're seeing thorium recoveries approximately 99%. And we're still evaluating the radionuclidic purity of the radium through this process. We are currently aging out the purified radium fractions to determine the underlying level of thorium-228 present in that. And at this point, we're about at the 500,000 decontamination factor, and we will still be assessing that over the next several weeks. In the second module, we prepared the radium for cation exchange absorption onto the generator column. At this point, the radium is progeny-free, so there's extremely low dose associated with it. And then in the third module, it's an automated column packing process where we homogeneously load the radium onto the cation exchange beads, and then form a slurry out of that and deliver that slurry and have it auto-pack a column. And we get very greater than 95% radium binding yield over this process. The other objective was to work this process chemistry into a fluidic system for each of the 3 modules that I just described, and then fully integrated into an inline end-to-end system. We have assembled this, what we're calling Generation 2 prototype system. It's been loaded into a glove box, or I'm sorry, a glove bag assembly, which is connected to the hot cell you can see in the background, and that's where the thorium-228 stockpile is kept and is prepared and aliquoted out for introduction into the system. The whole process from inserting the thorium stock in the front end to having a fully packed lead generator column takes right at an hour. And human intervention is limited to inserting the thorium stock into the front end and then disconnecting the generator system at the back end, and then basically loading it out of the third chamber of the glove bag for eventual direct shipping. This is what our generator column looks like. So how does it perform? Like I said, this is all very new data here based on 4 generator columns that we have prepared using the system in the glove bag, with the final one being about 3 millicuries in total activity. So once we have the prepared generator column, we transport it to another lab where we evaluate its performance, at least to the extent that we're able to in our laboratory. So once the generator is prepared, we milk it at 24-hour intervals. And we do these milkings using digital pumps. So we have very, very good control over the volumes that we're delivering, excuse me, through it, and also the flow rate that we're delivering through the column. We have been daily milking these generator columns for at least 10 days. And in some cases, we've gone up to about 14 days of milkings. And we're looking at several different approaches to the milking. One is to evaluate the coelution of bismuth and lead, and then the sequential bismuth removal followed by the lead elution. And then also a process whereby we are doing an inline lead-212 matrix conversion to acetate buffer. So the lead-212 is ultimately dispensed in acetate solution of about pH 6. Okay, so Roy showed a generator elution profile, and I believe if you had these side by side, this would look pretty similar to it. We get about 95% of the removable lead-212 off in the first 1 milliliter. Using 2 molar hydrochloric acid at a flow rate of 1 milliliter per minute. Looking at the most recent 2 generators side by side, these are the generators that had the most activity in them. We've done evaluations on the degree of breakthrough of radium-224 from the column in the lead-212 product. What we have observed on the left-hand side is that we have a radium breakthrough that is about at the, well, it's about 0.45% of the total activity is radium-224 in the lead product, 1 milliliter fraction. And then we evaluated the insertion of this very small disposable catch column, which is the same cation exchange material loaded into the generator column itself. It's a Lure connection, and we just add that to the outlet of it. And what we've observed is a pretty significant, roughly 10x reduction in the amount of radium breakthrough that we observe in these columns. There is a bit more scatter here, but I will note the circled items are values that were actually below our determination limit. So we're reporting our determination limit here. Okay, so how much lead do we get off in these over these 2 columns over repetitive milking cycles? So we have about 94% elution with the simple process. When we add the catch column, on these 2 generators, we've noted a bit more scatter on here, but the average lead-212 yield is about 91%. So we've also evaluated the we've looked at the elution profiles if we opt to remove the bismuth 212 prior to the lead-212. And so within this simple milking process prior to the 2 molar lead elution, we deliver 0.5 molar HCl, and you can see here these are immediate dose calibrator responses that we have a nice sharp bismuth 212 removal band here, followed by the 2 molar HCl elution band for the lead, which grows in. These are in elapsed days, so you can see that within about 0.2 days, roughly 4 hours, the bismuth here has substantially decreased, and the lead lead and bismuth at this point at 0.2 days have basically reached secular equilibrium. This is the same study, except using the disposable catch cartridge at the outlet. You can see it causes a very slight shift in the bismuth elution band, and it also causes a shift in the lead-212 band. However, we're still getting about the same fraction of lead-212 eluted in that 1 milliliter volume. Okay, so just a quick summary. With the new fluidic system, it starts with the thorium-228 stock solution inserted into the front end, and it ends with the fully packed generator column. The process takes about 1 hour, and it has a dramatic reduction in dose due to the staff members. The current low millicurie test generators appear to be performing good to date. This is still an ongoing study, and will continue into next year as we begin to increase the millicurie loading on these generator columns. Our thorium removal through our process is at least 500,000, and is looking like that will go substantially higher over time. We have about 90% lead yields in the 2 molar HCl. We've demonstrated elution bands looking at bismuth and lead sequential elutions. The radionuclidic purity using the disposable catch cartridge is about 0.04%. And we're also, although I didn't show any of this data, we're also evaluating a process to dispense the lead in sodium acetate buffer. And the benefit here, of course, is that you don't have to do offline processing of the HCl matrix lead components. So it saves a lot of time, and it saves a lot of dose, and reduces contamination risk, etc. if you can get the lead-212 off in a matrix that's more directly amenable to biomolecule labeling. So in FY22, we hope to scale this up, and as we go, we will continue to evaluate the milking performance of the generators, and also depending on how things go and how early in the year, we may seek end user evaluations, and this could be something that could conceivably be worked through the NIDC. So I'd like to acknowledge the DOE Isotope Program that supports this work, and would take, no, I guess I won't take questions until Roy gives permission, but so that's all I have. Thank you very much.
Thank you very much for that, Matt. Fantastic progress towards automating this system, and I'm sure we'll have some questions for you shortly. So with that, we'll enter our Q&A session, and looking at the Q&A board, the first question is it's for you, Mengshi, and it comes from Weigong. You may have read the question already, but the question is, what dosimetry have you used, and you collect radiation data?
I'm not sure what way is indicating for radiation data, but for the dosimetry in this preclinical model, we use the feed software like particle and heavy ion transport system, the software to calculate the S value, and then using the DigiMouse 3D voxel phantom model to calculate the geometry of the kidney in a 28 gram male CD1 mice. For that, we calculate the kidney dosimetry.
Thank you very much for that, Mengshi. I hope that answers the question. There's another one for you from Yushen Feng, who says, great presentation, Mengshi. From the animal biodistribution data, it seems the uptakes in large intestines increased during time. What do you think accounts for this? We
see this from time to time in rare cases. it could be the mouse eating the feces in the bedding, and also it could be some potential liver metabolism. That really depends on the amount of that potentially due to the amount of peptide injected. But we're doing some investigation on this.
Thank you very much, Mengshi. Next question, I believe, is for you, Rebecca. It's from Richard Wall. When lead-212 is injected, can you clarify how similar the distributions are of lead-212 and bismuth-212?
So we didn't do biodistribution in animals, but when they're freshly prepared before injections, and our radio TLC scans are taken right before injections. So we've eluted the bismuth out before the lead, and usually, I mean, with the long half-life, we wouldn't have much bismuth-212 in the injection solution.
That makes sense. Thank you, Rebecca. The next question is for Dr. Delpassand from Zhanghe. What is the interval time between the multiple treatments? How and what factors do you use to determine this interval time?
Well, our protocol calls for injection every 8 weeks. So basically 8 weeks plus or minus 1 week. Patients, they come back for the next injection. Of course, before each injection, blood tests, including CBC, metabolic panels, are done to make sure it's safe to give the next dose to the patients. But it is every 8 weeks, plus minus 1.
Thank you very much, Dr. Delpassand. And another one for you, Dr. Delpassand, from Hushan Yan. Which mutation for the 11-year-old boy?
Which mutation?
Which mutation for the 11-year-old boy?
I don't understand the question.
Yeah, I guess they're asking in your 11-year-old boy study, what was the mutation?
Well, this was a patient originally diagnosed at age 8 with pancreatic neuroendocrine cancer. As you know, overall neuroendocrine cancers are extremely rare, relatively rare. In pediatric population is even more, is rarer or less common. So this patient initially treated with 4 cycles of lutetium dotatate adjusted for his weight. And then I think about a year after that, we started seeing progression in this patient. We requested from FDA for a compassionate use of AlphaMedix. It was granted, then it was approved by the IRB. And so patient received 4 cycles, again, weight adjusted dose for this patient. And this is all I can tell about this patient. Patient is doing great right now. And, you know, this is, I thought, you know, this is a great thing to report that this works actually in a number 1 pediatric patients safely, and number 2 in a refractory settings as well. So to us, this was a great success, and we were very happy to be able to provide this to this patient.
Thank you very much. And then before we move on to a question from Richard, there's another one from Hushan as well that may be related, but they're asking, do you accept pancreatic cancer patients with Fanconi A mutation and KRAS mutation?
Here, basically, as I mentioned, and I think alluded in my presentation, we are looking at SSTR expression. As long as their baseline SSTR imaging study is positive, these patients can be enrolled, and we think they can benefit from this therapy. So the biology, as we know, I mean, in targeted radionuclide therapy is a lock and the key concept. As long as we can deliver the load of radiation to the site of the tumor using those receptors, we think we can be successful. So this is regardless of any mutation. This is regardless of any other proliferative index, KI index, or even primary site of the organ. And this is, I think this is a new concept that every day we believe more on that, and we feel that this is really the most important thing. You need to have a very specific target in radionuclide therapy and have a ligand that can specifically reach to that target. If you are successful in doing something like this, the results is going to be good regardless of any other parameters that we know. This is the way that this therapy works.
Thank you. And another one for you, Dr. Delpassand, from Richard Wall again. Any measurable alterations in renal function, i.e. not DLT, but perturbations of lab values for kidneys? Are you underdosing perhaps?
Great question, Richard. We had some decline in the renal function, but nothing clinically significant. And it appears to be also reversible. These patients are under follow-up and constant observation according to our protocol. But I have to mention all these patients also received kidney protective agent, amino acid infusion, starting about half an hour before the administration of investigational drug and continue for 4 hours, the typical protocol that we use for PRRT. But nothing significant. The rate of transitional decrease in some kidney parameters, nothing clinically significant. So it is very safe, and we haven't seen any kind of damage related to investigational trial. I think in our database, we have a couple of patients that they actually had sudden kidney, reducing kidney function, but they had different reasons. They had hydration, they had severe, for instance, diarrhea causing dehydration and led to temporary decrease in kidney function, but they were reversible. But as something that we can say related to investigational drug, we haven't seen anything so far.
Thank you very much. And then a question from an anonymous attendee. It may be Wei Gong again, but for you again, Dr. Delpassand. First of all, fantastic data. What do you attribute industry obsession with actinium to? Lead would seem to be clinically sufficient and perhaps the better choice.
Well, the selection of Lead-212, we had several reasons to go by this. And I think the most important one was the advance that we see by our business partner, RadioMedix, in pushing the technology forward, having DMF filed by FDA. So we thought it's a more mature and more advanced stage of having access to Lead-212 as an alpha emitter. So that was probably one of our main reasons. But also my personal bias is that probably, I mean, I can compare Lead-212 to an actinium, like a comparison between lutetium versus yttrium. I think actinium has 4 alphas, while Lead-212 has 2 alphas. I think we have to prove this. As I said, this is my bias, but I think we're going to deal with less toxicity when we are dealing with Lead-212 as compared to actinium. This is my bias. Improve hopefully in the future, but this is what I think is a probably appropriate analogy comparison of lutetium versus yttrium and Lead-212 versus actinium.
Thank you very much for answering that, Dr. Delpassand. Question now from Tyler Bailey. What safety considerations are taken into account regarding the 2.6 MeV gamma from the thallium-208 daughter, especially in the clinical setting? I'll probably have to defer to some of my more clinical-based panelists to answer that question, but just to provide a bit more information, then yes, working with the thorium-228 K chain is very radiation dose intensive. When we handle thorium-228 in larger quantities at Oak Ridge, we typically are handling it in hot cell facilities, shielded hot cell facilities. That provides provides protection against radiation dose to operators. We then, when we're preparing generators, for example, we work with very small batches of thorium-228 in a glove box, and we include local shielding wherever we can to help minimize worker dose per ALARA principles. But yes, the thallium-228 does have a very intense gamma ray associated with its decay. So I don't know if anyone from the clinical setting, Dr. Delpassand or Mengshi, if you want to provide any more context on how the thallium-228 daughter is handled in the clinical setting.
I can just make one comment. We are using thallium for obviously for imaging and dosimetry, and that has been helpful for us. For dosimetry, we use a medium energy collimator for doing the dosimetry. So that's all I can tell. It has been helpful for us for doing dosimetry.
Thank you, Dr. Delpassand.
Yeah, we usually put the LED generator behind the 6-inch LED shielding when we do the chemistry work. That's just the practice being integrated in our lab. That's just what we've been doing so far.
Thank you very
also,
much.
One thing for our localized shielding when we're handling the thallium is it's got an extremely high energy beta as well. So we always integrate a layer of plastic shielding in to ensure that we're minimizing the bremsstrahlung that's generated to the extent possible.
Thank you for that, Matt. I hope that helps answer your question, Tyler. The next question looks like it's directed towards you, Matt, and me. Is this new catch column used in a new batch of Lead-212 generator production next year? Well, I can probably say we'll try to still answer that, and we have yet to determine a time frame for implementing that chemistry. Does that sound about right, Matt?
Yeah, I think if it's something that that the end user would like to use, we can certainly include it. It's extremely easy to prepare and inexpensive, and we may even be able to find a commercially available cartridge that would essentially fulfill the purpose for it. So yeah, and the other thing I would like to evaluate in this coming year is perhaps alternative resins that may actually enhance the degree of radium capture through that transit as well.
Thank you for that, Matt. Mengshi, another question for you. Do you have clinical evidence for the renal damage during human trials of cancer therapy?
I think generally kidney is a late responding tissue for radiation. So from the clinical practice, the general approach to mitigate that is to do the co-infusion of a positive charged amino acid, as Dr. Delpassand mentioned. And the monitor of to monitor the kidney toxicity at later stage will be general glomerular filtration and blood tests like BUN and creatinine. The work we've done here is indicating that there might be some earlier phase biomarker in urine that can help us to design the dose and correlates with the toxicity, but that's just still very preliminary.
Thank you, Mengshi. Another one for you, Dr. Delpassand. First of all, thank you for your presentation. Do you have any other clinical practice using Lead-212 based ligand therapy for other types of cancer treatment besides PRRT and NET? Thank you.
We are working on that. We hope that we have new agents to treat conditions such as GBM of the brain and some other conditions such as ovarian cancer or even prostate cancer. So I think the menu on these agents will only increase. And hopefully, I mean, with the results that we have on the NET side, we can use our experience in this area for treating other unmet conditions that we are dealing right now in the clinic. So to me, the diseases that I mentioned, they have the highest priority.
Thank you very much for that. Another one for you, Dr. Delpassand. What did the NET test from Ren Laboratories show?
I defer this question actually. It showed in majority of the cases, it showed correlation with our response to therapy. I think we had a few cases that the results was not quite fitting the results of the NET test, but apparently, you know, there are some maybe medications or that can affect on the results of this test. But I think in the majority of the cases, actually, we had correlation with the response to therapy. This is what I have. We hopefully have more detailed information and publication related to our clinical trial that maybe we can in the future we can report that. But I can tell you the short answer is in the majority of the cases there were actually correlation between the test and response to therapy.
Great, thank you. Question for the panel here from Larissa Viola. Is the presence of bismuth-212 critical in the injected lead-212 dose, or is it okay to have a mix of radioisotopes? Well, hopefully one of the panelists can elaborate on that a little bit better than me, but I think it's absolutely fine to have a mix because we're looking for the bismuth-212 to do the work anyway. Anyone else want to elaborate on that?
I can provide some some of the based on our data, the dosimetry data, the alpha particle actually contribute to up to 93% of the total dose, whereas the the electrons were slightly above 7%. So the decay, the alpha particle generated from the downstream of bismuth is actually quite important. But that really depends on the ligand itself, right? The PK of the ligand, the biological half-life of the ligand, whether it's the antibody or it's a peptide or it's a small molecule. And in our case, we in the radiochemistry process, we purified the lead-212 from all the others radioisotopes. But I mean, for sure, the bismuth-212 will grow in with a half-life of 1 hour. So again, it depends on the targeting vector itself, and the tumor retention and how fast it can clear through the non off-target organs.
Thank you very much for that, Mengshi. Another question for you, Dr. Delpassand, from all here. What is the dose to the kidney? Have you seen changes in creatinine clearance, and how long is the follow-up? Given that it maybe given that it may take time to see toxicity?
Well, I think I've kind of answered this question. We haven't seen any clinically significant deterioration of kidney function in our patients that has been related to the investigational drug. We have follow-up in some of our patients, more than 2 years after the first injection. So, of course, all these patients will continue follow-up for as long as they are alive, actually, because one of our things that we want to actually evaluate is overall survival. So we'll see if there is any long-term effect on these patients. But I mean, to me as a clinician, when I see these patients with a stage 4 disease that they have really, I mean, they have used a lot of medications in the past. They come for this kind of a therapy. The safety profile is not one of their concern, and clinically also not ours. Our experience, as I told you, in more than 1,000 patients I've treated with PRRT, these are, as long as you use the protective agent, this is basically the standard protocol. This shouldn't be a huge concern for these patients with this end-stage disease, number one. But I can tell you that so far, we haven't seen, in some patients with more than 2 years follow-up, anything really significant, clinically significant as it relates to the function.
Thank you very much, Dr. Delpassand again. Next questions are from Rachel Hausman. The question is, I saw her no mention of NIDC production of lead-212. How much are they making? Do they have an estimate for demand and their capacity? I think that question, Rachel, will have to be directed to NIDC directly. Probably best to shoot them an email on that one. I'm sure they'll be happy to answer that for you. Next question is for the panel from Richard Zimmerman. Lead-212 is the first beta emitter and only decay radionuclide, bismuth, polonium, or alpha emitters. Would you consider lead-212 being an alternative to co-injecting lutetium-177 and actinium-225, which could explain a potential higher efficacy?
Is this
any of the panelists?
Well, I can take this. I mean, this concept of tandem therapy has been discussed in nuclear medicine for many years. I mean, theoretically, it makes sense in this way that, okay, these lutetium has a different path length in the tissue, alpha emitters, they have a different path length. And so by having isotopes with different path lengths and energies, you can target the smaller lesions, bigger lesions, etc. Overall, again, my personal bias is that anywhere that there is a blood flow to the tumor, we should be able to reach that. And the more effect comes from the energy of the particles. And this is why all the time, theoretically, we were thinking that the higher the linear energy transfer, the more damage to the DNA, and especially double-strand DNA damage, which should give us a better efficacy and longer time to progression. All these ideas were sort of theoretical until we actually put this in the patients, and now we are seeing in practice in our clinic how they respond. And we see the progression-free time for beta emitters, and now we are started seeing alpha emitter effect and see how different is that. So actually, we have been able to realize all those hypothetical things that we were talking about in actual patients, which is obviously extremely exciting. Combination of these drugs given to the same patient, to me, is going to be on the regulatory side, and drug development might not be that easy. I mean, looking at different dosimetries in the patients, and okay, if you are dealing with the side effect, where it's coming from, I don't know. I mean, I feel it's going to be challenging in terms of regulatory aspect and drug development, although scientifically it's intriguing to see really these tandem therapies will help us in terms of better efficacy and safety. But if we have one agent that does the job, of course, I think that's all we need in the clinic. But overall, the main problem and challenge that I see with tandem therapy is on the regulatory aspect, and taking it through regulatory agencies such as FDA or EMA. It's going to be a lot more complex approval of these drugs that uses different type of isotopes in the same setting and in the same patient.
Thank you very much for that, Dr. Delpassand. The next question is from Jorge. And the question is there any data using NGAL, TIMP2, IGF-BP7 in renal damage from radio-labeled somatostatin analogs?
I think this question for me. Actually, there is. I just came across with a paper on a preclinical study using the bismuth-213 dotatate in this study, the authors look at the Abergel to as a biomarker to determine the kidney toxicity and the how amino acid co-infusion help to help the kidney toxicity. But in that study, the authors look at the time point at 3 months post-injection, whereas the Abergel could be a more sensitive biomarker at the earlier phase for acute kidney injury.
Thank you very much, Mengshi. Another question for you, Dr. Delpassand, from DG, who's from ViewPoint Molecular Targeting, and the question is whether 4 treatments are sufficient or overdosing, and how does it relate to toxicity?
Well, we have proven so far that it is safe. We haven't seen any toxicity for administration of 4 doses, and in fact, we haven't reached maximum tolerated dose, if you will. I mean, still probably patients then they can receive, but since we are seeing such a great response, I don't think we need to push the boundary in here and like chemotherapy agents to necessarily find the MTD dose for this. At this point, with this current therapy and with this current dose, we are getting the response that we want, and with the extremely safe condition for our patients. So I think if it's not, why scratching?
Thank you, Dr. Delpassand. Another question for the panel here, and first of all, thanking the speakers. And what do you think the main challenges for Lead-212 clinical developments in cancer therapy are? The generator side or the impact from actinium-225? It seems that more companies are working on actinium-225 with funding penetration as well. Is there any research on Lead-212 PSMA or Lead-212 FAPI?
I didn't, I don't know, it was breaking up. I didn't get the first part of the question, but if the question is, are we developing anything related to prostate cancer or FAPI? These are all great areas. FAPI is a great molecule also, yet to be seen how, in terms of, because essentially the affinity is for fibroblasts, and this is not necessarily cancer cells. How you can, and we know alpha emitters, they have a very short path length that can be, by just being close enough to the cancer cells, can we insert the therapeutic effect of an alpha therapy? I think this is yet to be seen. But in answering the question that if we are using it in other setting and other unmet needs in oncology, the answer is yes. And this is a very exciting area. I think all of us that we are working in Lead-212, we need to really looking at these unmet areas and try to develop drugs that can help our patients with these conditions.
Thank you again, Dr. Delpassand. And there's one final question for you, and that's from Jorge again. What is estimated dose to the kidneys and to the tumor?
I think we're going to have a publication. I don't actually, I can't tell you from the top of my head, but we'll have a publication dedicated to dosimetry for Lead-212, labeled Temptate. So stay tuned. We're going to publish that.
Thank you very much.
Thank you.
I think there was one more. I thought we were done on questions, but I think there are some more questions that were submitted to the chat thread. Pull these out. There's one here. This is a comment and a question for Dr. Delpassand. Amazing results and congratulations for a study well done. Do you have any dosimetry from your studies, both tumor and normal organs, to see if they're in line with the responses and the safety profile?
We have done dosimetry on 6 subjects based in this phase 1 trial. And as I mentioned, we'll have publication on that. The dosimetry is favorable. And I think regardless of numbers, I think the picture is worth 1,000 words, and you saw the effectiveness. So that we do have effectiveness on the tumor, and the safety studies that we have so far in patients suggest that it is safe in our patients. So far, we'll continue doing all the safety studies down the road. But we will publish the exact numbers from our dosimetry studies, hopefully in the very near future, with the help of our dosimetrists at Rapid.
Thank you very much, Dr. Delpassand. And with that, I think this concludes the Lead-212 user meeting for this year. Thank you all for participating in it. Thank you all to all the panelists for some fantastic talks and for accommodating all the questions at the end of it. A note to let you know that this is being recorded. I think it will be available on the DOE Isotope Program website sometime soon. So look out for that. And thank you all very much again. Enjoy the rest of your day.